Hi, everybody. Hi, everybody. Welcome to WMS Lecture. We have our lectures every third Thursday. So if you forget, just remember, it's Thursday. Is it the third one? You know there's going to be a lecture. And so we have our lectures every third Thursday. We start 6:30 with our pre-lecture and talk a little bit about what people are finding. And then we usually go into our lecture about 7 o'clock. Tonight, our presentation is entitled The Joy of Fungal Sex by Patrick Leacock. This presentation looks at the strange and wonderful diversity of reproductive strategies and life cycle options. Ever since fungi arrived on the land and developed hyphae, they have explored ways to propagate, soon rejecting a basic male-female sex paradigm. Fusion of compatible hyphae controlled by mating genes replaced the use of single-celled gametes. I'm sorry, can someone watch the admissions? Excuse me. Some fungi have two mating types. Other fungi have hundreds or even thousands. Many fungi, such as molds, reproduce asexually. But when the going gets tough, some fungi can self-reproduce sexually. Mushrooms have gone a step further to keep their options open. Rather than cells having a diploid nucleus following mating, the nuclei from the two mated individuals remain separate, forming a dikaryon, which with two haploid nuclei in each cell. This allows a mated individual to fuse with and fertilize an unmated individual by donating compatible nuclei. Oh, and those mushrooms that we love to collect and cook for dinner, those are spore-producing sex organs. So it's kind of, like sweet treats, right? Anyways, I'd like to talk a little bit about Patrick Leacock. He's a mycologist documenting the mushrooms of the Chicago region with collections going to the Field Museum of the Natural History, where he has worked for 14 years and continues on there as an associate. He now is at the School of the Art Institute of Chicago, and he assists on forays as a scientific advisor for the Illinois Mycological Association, and he's also a scientific advisor for the Wisconsin Mycological Association. He started his mushroom activities with the Minnesota Mycological Society and obtained his PhD from the University of Minnesota. Patrick served as a voucher coordinator for 20 years, documenting fungi at the North American Mycological Association annual forays. And as you can see, his website is amazing. And definitely take a look in the chat at all the great links that are there for you, especially the Myco Guide chat. And with that, I'm extremely proud and excited to introduce to you Patrick Leacock.
Thank you. Let's see if I can share my screen here. So I've given this talk to a number of clubs. It's been in development for the last couple of years. And coincidentally, I'm in the middle, or this is the first week of a 3-week summer session class. It's a full semester class squeezed into 15 days in 3 weeks. So today, tomorrow, and Monday, I'm actually doing these. The students are getting my life cycle talks. So they get 3 lectures and you're getting 1. So it's boiled down into a more condensed lecture with the high points without as much detail. So if you have questions, we can have those at the end after I go through the PowerPoint. And just 1 second, sorry. So fungus life cycles are still being unraveled and figured out, but they're very interesting. And this is a John Raper quote from a while ago. Sexuality in fungi or fungi, however you want to say that, has long been recognized as one of the more perplexing yet intriguing facets of the biology of this large and varied group of microorganisms. So I'm going to compare fungus life cycles to plants and animals for the first part. And one important difference between those three groups of complex life forms is that fungi don't have tissues like plants and animals. So we're more familiar with animals and they have a body plan. There's usually a symmetrical arrangement of body parts, and an individual might grow larger, but the body parts don't change over time unless you have like an insect that goes through a metamorphosis or whatever. But there's usually a set arrangement of body parts. Plants are what I like to think of as modular. You can have a bunch of stems or just one stem. You can have a bunch of leaves, any number of leaves, but some plants just have 2 leaves, and you can have a bunch of roots. There usually aren't a set number of parts, stems, leaves, and roots, and flowers and things. So plants are modular. Stems can make leaves and flowers, and roots can make more roots and things like that. So they have these parts or organs, if you want to call them that make up the whole plant, but there's no defined structure. There's growth patterns that make different shapes of trees and so forth, but they're not like all the same exact branching pattern, for example. Fungi are mostly single cells or filaments of cells in a thread called a hypha. And any mushroom structure, any fungus structure is going to be made up of these threads unless it's a single cell fungus. So a mushroom fruit body like a bolete, if you tear it and open it up and look at it under a microscope, it's basically a lot of threads all fused together. And then at the tips of the threads, you have terminal cells of different types. They might be what we call cystidia, which are the terminal sterile cells, or you have basidia and other kinds of cells in there, but it's all based on a filamentous branching structure. There's no true tissue where the cells divide sideways. They only grow at the tip and branch. They don't make a 3-dimensional tissue from cell division. It's by grouping filaments together to make a shape that's more organized. So the growing part of the fungus is the mycelium, and that's down in the soil or in the log, and that's the threads of cells with cross walls to make separate cells in that thread or the hypha, and a whole bunch of hypha together make a mycelium. And then the part of the life cycle that we like to look at are the mushrooms, which are the fruiting bodies or the reproductive structures, sexual structures of the fungus. And they have the temporary job of making spores. So a lot of mushrooms don't stick around very long because they're more like flowers or fruits or whatever. They're there for a shorter time to make spores. One way that fungi reproduce that we use for culturing, like Tavis does when he grows mushrooms, is fragmentation. And that's where the mycelium breaks apart, either when there's disturbance or something, or if we go in and we take a piece of a mushroom or piece of a mycelium and stick it on another agar plate, that's a fragment of that individual, and that can grow into another individual. So this is, in a way, it's like cloning. You're just duplicating that individual by separating fragments and having those fragments grow into a new individual. Lichens also do this. They have fragments. And I don't know if you remember from high school biology or way back, there's two types of nuclear division. There's mitosis and meiosis. Mitosis is the regular copying of DNA. The chromosomes are duplicated and then separated into what we used to call two daughter cells. I don't know, or two sibling cells. So that if you have a diploid nucleus with two sets of chromosomes, after mitosis, you have two diploid nuclei. You end up with the same number of chromosomes as they're duplicated and then separated into nuclei that are put in two different cells. If you start with a haploid cell, you're going to get two haploid cells. So mitosis is cell division where you're just ending up with the same kind of cell components, the same genetic makeup as the first cell. Meiosis is part of sexual reproduction where you have a diploid nucleus, two sets of chromosomes. Those are duplicated, and then there's two rounds of division that
Maybe.
separate those chromosomes into four.
Coffee, it's too.
We've seen the talk before.
Can everyone mute, please?
You end up with four haploid nuclei that are then, in the case of mushrooms, separated into spores that are haploid. So mitosis is found in lots of places in the life cycle. Meiosis only occurs where you're going from diploid to haploid. Plants and animals have what we call gametes, sperm and egg cells. And this kind of male-female sperm and egg type thing is based on animal life cycles. And we sort of applied it to plants where it's more obvious where part of the plant is moving from one place to another and carrying nuclei, whereas the other part is sedentary and stays locked away inside the flower. So we've called the pollen grain there on the bottom right that moves around from one flower to another carries nuclei in there. So we call those sperm nuclei because they function like sperm do in animals, and they fertilize what we call the egg cells in the ovule in the flower. So it's an analogous kind of part of the life cycle where haploid nuclei of one individual are fusing with haploid nuclei of another. And in the case where that nucleus is bound up in a little cell and moves, then we tend to call it a sperm if it has a way to move over to where there's a bigger cell that it mates with. It's usually called like an egg. But in fungi, we don't usually have that kind of dimorphic difference in morphology and function. So some of the lower fungi, simpler fungi that are microscopic, they have, we might call them gametes, but they're zoospores, which are spores or single cells with flagella. They swim around and then fuse, and those act like gametes because they are fusing with other cells. So their function is like gametes, but there's not usually any clear male-female assignment there because they both look the same. So there's different ways that sexual reproduction is beneficial. Meiosis provides some rearrangements of chromosomes, including crossing over events where you get genes mixed between the chromosomes. This gives you genetic variation in the offspring, which lets them adapt over time to changes in the environment. And then there's different organisms might have different rates of genetic change over time. So some lineages are changing faster than other lineages. This can cause a problem when you're comparing sequences. So as a base point or starting point, we're going to look at a life cycle of an animal. So this is a generic animal. And we're going to start out with what we call a diploid dominant life cycle. So this life cycle is the diploid organism there, that animal lives most of its life with cells that have two copies of chromosomes. That's what we have. And then at some point in the life cycle, it makes gametes that are little haploid cells. But the organism lives most of its time as a diploid with lots of cells. It's called multicellular. There's two processes to switch between the two halves of the life cycle. So to go from diploid to haploid, like I said, there's meiosis. That's a reduction division to go from two sets of chromosomes to one set of chromosomes. And then to go the other direction, you have to have two gametes or equivalent things fused together in what we call fertilization to go from haploid back over to the diploid, where we have one set of chromosomes joining with another cell to get two sets of chromosomes. So we have these arrows here to go circle around in the life cycle and go and switch back from diploid to haploid and back to diploid. And on the bottom, we have meiosis for diploid cells. And when a diploid nucleus divides by meiosis, we get four products, four haploid nuclei. So that's why I've shown four cells here, four gametes in this case. And then two of those gametes, sperm and egg, would fertilize, go through fertilization to make the zygote. The zygote grows into the animal, adult animal. So this is the diploid dominant life cycle. It's dominated by the diploid individual. The only haploid part of the life cycle are the gametes. So the haploid side is simple, the diploid side is complex and long-lived. So in fungi, they undergo mitosis and meiosis just the same. Mitosis, like other things, is for growth and making more cells. Yeasts are single-celled algae, and they divide by mitosis. They can also have a sexual cycle. So some organisms use mitosis to make little cells that bud off, and is a method of asexual reproduction where it's just making more copies of that same individual by mitosis. So there's no change in the genetic makeup or the chromosomes. It's just making more cells, and the cells separate to make new individuals. So that's not, so it's asexual. There's no fertilization and meiosis involved in that process. So yeasts are a common example of asexual fungus, and this brewer's or baker's yeast, Saccharomyces, has budding method of making new cells. In this type of reproduction, mitosis, you have the original cell starts a little growth off of one little spot, and that growth gets bigger and bigger, grows bigger and bigger, and then separates off. So we call that a bud. It's a second cell that grows off of the first cell and gets bigger in size until it's mature, and then there's a cross wall that forms, and it breaks off. And that cell on the top right, you can see, has budded at least 4 times. There's scars left on the cell wall each time it forms a bud. So that's a basic yeast. The other type of yeast that are not as common is the fission yeast, and they divide more like bacteria, where the cell gets grows in length and then splits in half. It grows in length, gets longer, and there's a new cross wall laid down in between, and then it separates into 2 cells. So it's not like the cell is making a new cell on one part of it. It's the whole cell is getting bigger and then splitting in half.
Press.
So let's build a yeast life cycle. And to do that, we're going to start with a basic life cycle, like we've seen, where we go from zygote or diploid to haploid and back again. But a yeast cells are all single celled, so there's no multicellular part of the life cycle. So both sides we have unicellular single cell yeast cells. So part of the yeast life cycle is haploid. I think it spends most of its time as haploid, and then it can go through fertilization to get diploid, and then go through meiosis to make spores that then germinate back into the haploid yeast cell again. But yeasts mainly grow by mitosis, like when you're brewing beer or baking bread or having the bread rise. You have all these yeast cells that are duplicating really fast as they eat up the sugar, and they make carbon dioxide and all that. So they're replicating their cells. This is just asexual reproduction, rapid cell division. And it can do that with either the haploid stage or the diploid stage. So we're going to add these two cycles onto our sexual cycle, and we get this, where we have the sexual cycle in the center, and then both sides, right and left, we can have that diploid or haploid cell go through the asexual cycle of buddy to make more cells. So, and one thing I found looking at yeast life cycles is the fertilization stage where two yeast cells fuse together is called schmooing. So you can see that, whoops, I took out that other slide, but that's, I don't know where that word comes from, but that's called schmooing, where two cells come together in a yeast. So there's three basic types of life cycles, and then different organisms have variations on these. But diploid dominant here is where the, as in animals, the diploid organism is dominant. It spends most of its life as a diploid. And then the mushroom life cycle has a variation on diploid that we call a dikaryon. It's got two sets of chromosomes, but those are in separate nuclei. So on the top right, we've got N plus N. That's a cell with two nuclei that are, the two nuclei are haploid, but those come from the two parents. And I'll explain that in a minute. And then some life cycles are haploid dominant, where it spends all its life, most of its life as haploid. And then plants and some other things have an alternation of generations where there's actually part of the life cycle spent either diploid or haploid. It has spent some time in both stages. And then a lot of things have asexual life cycles, which are a different way, different methods. So there's three different nuclear conditions you can find in fungus cells here. You can have diploid nuclei, and that's found in honeycaps and a few other exceptions. You can have haploid nuclei that's in molds and a bunch of hyphae before they mate, they're haploid. And then this dikaryon stage where you have two nuclei from each parent, a nucleus from each parent in the same cell, but the nuclei are kept separate. That's dikaryon, that means two nuclei. That you find in mushrooms and the ascomycetes. So it's in the basidios and the ascomycetes. They're like regular mushrooms and morels and cup fungi and carbonaceous fungi. The animal life cycle, just to reference here, is diploid dominant. Most of life is spent as diploid on the right. Here's haploid dominant. The only diploid stage is the zygote after fertilization, and that, in many cases, that's a resting stage. And like in some of the algae, the zygote has a thick wall and allows it to live over the winter and stuff in the pond. But most of its life is spent as a haploid individual. So a lot of algae are in this kind of life cycle. This is what plants have, and some fungi have an alternation of generations. They switch between a haploid individual and a diploid individual. And we'll look at that briefly in plants. There's somebody that needs to mute. That's not me moving stuff around.
Hi, that's Anne.
Yes.
Sorry, you need to mute. Also, I'm sorry if my audio is doing something weird. It sounds like it is to me.
That's feedback.
Thank you.
This is a simple tree of the different fungus groups, and it's showing the simpler fungi up on top. The chytrids, zygomycetes, and glomeromycota are three groups of microscopic fungi. They're not things that we collect when we're on forays. They're down in the soil or in the pond. And what we do collect on forays are in the two groups on the lower part, basidiomycetes. Most of the mushrooms are there, and the ascomycetes has cup fungi, morels, carbon fungi like dead man's fingers and some of the plant diseases and stuff down there. But the Basidiomycota and the Ascomycota are two groups that have this dikaryon stage. So they're different from the other fungi by having this pretty much unique method of keeping the two nuclei separate in the same cell. Some of you know mushrooms have lots of different kinds of spores, and fungus spores vary greatly in size, especially in ascomycetes. But the basidiomycete mushroom spores are mostly fairly small, 5 to 10 or 15 micrometers. So some of the small spores are shown there in the bottom with the Leacaria and the Ganoderma there in comparison to some other fungi. And then the orchid seed there on the top right. Molds have asexual spores. We call them conidia. That's just a term for these asexual spores that are budded off of these branches. And two common types of mold are penicillium or blue mold and an aspergillus type. I think those are brown or black. There's also another black, like bread mold, that is in a different group. That's in the zygomycete group. But these are asexual spores. Here's mushrooms, and these are in the Basidiomycota, and these are reproductive structures, and they're going to make spores on the particular surface. One thing that's unique in the Basidiomycota are these clamp connections. So if you get to identifying stuff under the microscope, one of the things you look for is clamps, because certain species of mushrooms have clamps and some don't have them. But if you see clamps, then you know it's a Basidiomycete fungus. It's not in the ascomycetes. And it's thought to have, one function of this is thought to be to help these nuclei divide. Like in the diagrams there, the cell has two nuclei, one from each parent, and they divide synchronously when it makes a new cell. So one nucleus divides into this clamp. There's a little branch that goes backwards, and then one nucleus divides up into the branch, and the other nucleus divides along the main axis. And then the separated nuclei are then end up on two sides of the cell, and there's a cross wall there in the filament and in the clamp to separate those two parts of the hyphae into two cells. And then the hypha keeps growing at the tip, and then when it gets long enough, it'll divide and make another clamp. And on the bottom is what clamps look like under the microscope. Sometimes they're hard to find, sometimes they're obvious. Here's mushrooms and Basidiomycota. So these spores are made on the gills or the tubes, or on clubs, or on teeth, or on just a crust, like a crust fungus in the lower right there, where it's just a surface, spore-producing surface on the log. That's really simple form. And then the basidiospores in a basidiomycete fungus are produced on the outside of the basidium, which is the reproductive cell. And those spores get shot off. And on the bottom there are drawings of a bunch of different kinds of mushroom spores. So that's one thing that's fun about looking at things under a microscope is the spores have a lot of variation in both color and size and texture and ornamentation. That labeled B there, that's some of the Russelolacterius have really big warts. Inocebees have, some of the Inocebees have bumpy things. Letter A that's Entoloma type spores. G, I think, is Lepiota, some certain Lepiota. And then R is more like a Leacaria spore. So there's variation there in the spores. This basidium is the sexual spore for where meiosis occurs, but also where karyogamy occurs first. So on the left there, we have the dikaryon stage hypha, and it makes a terminal cell, the early basidium cell. And at number 2, we see it's got 2 nuclei, one from each parent. And then we've got karyogamy where the 2 nuclei fuse, that's at number 3. And then at number 4, we have meiosis where that nucleus divides by reduction division to end up with 4 nuclei. And then that basidium starts making spores on the outside on the top. And number 5 is a vacuole of water that is kind of pumped in and helps push the cell contents of the basidium up into those spores. And the nuclei migrate up through those stalks at number 6. And then at 7, you end up with the spores, mature spores, each one there with 1 haploid nucleus. This shows the spore discharge, which isn't completely understood, but part of the process is this Buller's drop, which eventually people figured out was just water. It wasn't any mystery liquid, it was just water. That's water that's exuded out of the spore at the base and gets bigger. This happens when the spore is mature, full size. That water droplet grows bigger, and then when it gets big enough, it'll come into contact with the liquid that's on the spore, and that water then moves up onto the spore, and that changes the center of gravity for the spore, and that makes the spore break off of the stalk. And then the hypothesis I like is that the negative charge of this whole structure makes the negative spore repelled away from the negative basidium, because they have the same charge, and then when that breaks, the like charges repel each other. There's probably a Wikipedia page that explains this in more detail. Most mushrooms have a regular basidium with usually 4 spores. In jelly fungi, there's actually 4 different groups of jelly fungi, and they have different basidia. The tree ear, wood ear there, Auricularia, has a basidium that's split horizontally into different cells. The tremella and things like that, and Subocina has a vertically divided basidium. And the Dacromyces jelly has what they call a tuning fork. It's not split, but it has these 2 branches that are long. And so you only have 2 spores on that one. The interesting thing about jellies, well, jellies are hard to look at under the microscope because that jelly layer is really strong. And the basidia and other cells are embedded down in that gel. And the strigma, the stalks of the basidium, extend up, like in the top right drawing. They extend up through the gel to the surface, and they make the spores in the air on the surface of the jelly layer. So let's finally make a mushroom life cycle. So here's a haploid dominant life cycle. And we're going to get rid of sperm and egg gametes. We're going to have haploid nuclei that come together, but we're going to not have actual single cells that mate or fuse. It's going to be the hyphae instead. And then fertilization, we're going to split that into 2 steps separate in time. And these 2 stages can be separated by a lot of time. So we're going to crack open that fertilization, make it into 2 parts here. So fertilization is actually 2 processes usually happening right after the other. Like when a sperm fertilizes an egg, what happens is first that sperm cell fuses with the egg cell, and the contents of the sperm cell get dumped into the egg cell. And then that nucleus from the sperm moves over and then fuses with the nucleus of the egg. So that's 2 separate things. So the plasmagamy up on the top is the 2 cells fusing together, and the karyogamy is the fusion of the 2 nuclei. So what we get in mushrooms is actually a separation of those 2 processes in time. So we get this new life stage in between that of this dikaryon. So we get plasmogamy, we get mating and plasmogamy where two individuals come together and fuse, but the nuclei from each individual end up in the same cell, but they don't fuse together. So we get those two separate nuclei in each cell. So that's the dikaryon stage, which is new for the basidiomycetes and the ascomycetes. And that can last a few hours or days or weeks or years or decades or thousands of years. So like the big armillaria colonies up in northern Michigan, they found it's 37 acres, and out west it's even bigger. Those have been living for thousands of years. We don't really know how long, but one individual keeps growing and getting bigger and bigger over time. But in actual case, a lot of armillaria are actually diploid. I don't know if it's known for each species, but some armillaria species are actually diploid rather than dikaryon, but they can live for thousands of years if they don't run out of food. So anyway, we have this fertilization in 2 steps here. So if we take our basidiomycete life cycle and look at a particular organism might have other things it can do besides the basic sexual life cycle. So the basic life cycle, sexual cycle is in that box, the lower right part. And then this Coprinus, this Coprinopsis cinerea, which is a inky cap type mushroom, it has 3 different ways it can reproduce asexually, spread itself around without going through the sexual stage. So when this fungus wants to reproduce sexually, it has to go through the karyogamy and meiosis inside of a mushroom to make new spores. So it has to go through that part of making a mushroom. But the mycelium of either the haploid or the dikaryon stage growing in the soil or wherever has options on reproducing itself without sex. And the oidium stage on the top left there is a simple method of just making little spores that bud off. And these are just clones. It's similar to what molds do. They just bud off little cells. But this fungus can also have 2 different kinds of resting stages. The chlamydospore is a single cell that has a thick wall. And the sclerotium is a bunch of cells in a tight mass with a lot of energy storage. So it's a larger structure to store energy and survive like over the winter or stuff. And morels also makes sclerotia, and some other mushrooms makes sclerotia. So if you look at a particular species of mushroom, it might have typically the basic life cycle for sex, but then it might have a couple ways to have asexual reproduction. But some mushrooms out there, some of the mushrooms we collect, don't have any known asexual stage. So they have to go through a sexual stage of making mushrooms in order to reproduce. So one thing I've been working on recently is these life cycle quiz handout puzzle thingies, not sure what to call them. And I plan to put this on my website, but they're not up there yet because it's not finished, and I need to write up a description of what all this means. It's kind of a lot of little cartoons of fungus things going on here. But we'll go through kind of quick an example of a mushroom life cycle using my little puzzle pieces here. So the idea is you get a PDF and you print that on like some stiff paper if you can, and then cut out each of the rectangles, and then you have to piece those together into the life cycle that we just showed. And I'm adding variations for different mushrooms like the honeycap and whatever, and different types of spore production and things like that. So I've got the basic life cycle done, but I'm still adding some variations to that. So this is an example of the basic mushroom life cycle. These little labels all come on one page, and you have to cut up all these little rectangles. But we're going to take all of this and piece it into a life cycle. This is the more complex form where every little bit is separate. The labels and the arrows are separate from the diagrams of the fungus, so it makes it a little more challenging. But we're going to start out with the spores. So we've got a spore on the top center there. It's got 2 nuclei, so we call it binucleate. I'm just explaining the terms put on here for people that like to see all these terms. The spore has 2 nuclei, but they're the same. So it's called a homokaryon because both nuclei are the same. Anyway, that spore germinates, starts making thread cells or hyphae by mitosis. That grows into a haploid individual. And that haploid individual is a monokaryon because there's only one kind of nucleus there. And if it's lucky, it finds a different mating type in the soil or in the log. And I'm showing different mating types by different colors of the nuclei. So the fungus on the right finds a compatible partner in this other mycelium in the center. And they can grow towards each other and find each other and then fuse. And then the nuclei can come together into the same cell and start making the dikaryon. So this shows how that happens. So in the top left, we've got those 2 individuals coming in contact with each other in the top right there. In the lower right, those 2 hyphae fuse, and we get a cell there in the middle that has both nuclei in it. And then a miracle happens where the existing monokaryon or haploid individual can now duplicate the new donated nucleus from the other individual. And those copied nuclei then can propagate back through the existing cells and make all the cells, at least in the vicinity, can turn all the cells into a dikaryon. It's not just the new growing tip. But we have a new growing tip there in the bottom left that's growing out with clamps because you only have clamps with a dikaryon. And that is going to grow into more dikaryotic hyphae. So we're now at the middle right there with a dikaryon with clamps. That is going to grow. So that's a mated individual. That's not it's a dikaryon. It's got 2 sets of chromosomes there. It's going to grow by mitosis into a bigger and bigger mycelium. So all those cells in the mycelium are going to have the 2 nuclei. And then at some point, it's going to grow by mitosis. Those hyphae are going to grow and make a mushroom. So that mushroom shape is just made out of hyphal strands, which are very organized into that shape. Whoops. And then on part of that mushroom, in this example, the gills, we have the hymenium, which is the fertile layer. It's the layer exposed to the air that's going to make spores. So the mushroom grows by mitosis to make all the different parts of the mushroom. And there's these terminal cells on the hyphae, on the gill, that are going to make the basidium. And in the bottom left there, we have the new basidium in the dikaryotic condition where we've got 2 different nuclei there. So this is called a probasidium. It's before karyogamy. So now we get karyogamy and the steps of sexual reproduction here, going from the bottom to the top left. So we have karyogamy, which those 2 nuclei fuse together to make a diploid nucleus. You can call it the zygote if you want to make it analogous to other life cycles. And then that diploid nucleus divides by meiosis to make 4 haploid nuclei. So meiosis always makes 4 products. So you go from 1 diploid nucleus to 4 haploid nuclei. And then those 4 haploid nuclei are pushed into the spores that develop on the outside of the basidium. And then in this particular example, which are nuclear patterns D and F, in this case, we're looking at pattern D. We end up with what's called a post-meiotic. It's not labeled here, but there's what's called a post-meiotic mitosis. So we have 4 nuclei from the meiosis. Those 4 nuclei, one each goes into each spore, and then those nuclei divide after they go in the spore. So we end up with the spore with 2 nuclei. So this is what we find in some of the mushrooms where we have the spores have 2 nuclei in there from the original condition when they were shot off of the basidium. But there's different patterns of how these nuclei divide when the spores are made, which it's very complicated for me to research and figure out what the different patterns are, and it's even harder to find out what the examples of different mushrooms are. So that's part of what I'm going to end up putting on the website is examples of how different ones do this differently in the reproduction. So this is a chart that's going to be on my website with an explanation. I'm not going to go into detail here, but there's different patterns, A through F, on how the nuclei divide and move, and whether they divide and then move, or whether they move and then divide, and then they move back again. So for an example, in the on the top, we have 8 nuclei. Top center, we have 8 nuclei. Those move into the spores in the top right, but on the other one, pattern A, only 4 nuclei move into spores. 4 nuclei stay behind and aren't used for anything. Down and below, in some other examples, we have the nuclei go into the spore and then divide, and then some of the nuclei migrate back out of the spore. So it's kind of strange. And then in the bottom left, there is what the store mushroom is doing, Agaricus bitorquis versus, Agaricus bisporus on the bottom is the store mushroom, and then Agaricus bitorquis is more of a regular Agaricus that has normal spores, but the Agaricus bisporus makes 2 spores, and each spore has both nuclei. So these spores, when they germinate, are already a dikaryon. They don't have to go through any mating, which is kind of cool. The other neat thing is a dikaryon on the top left there can mate or fuse with a haploid individual and donate a nucleus to the haploid individual in the equivalence of mating. So that haploid individual gets a compatible nucleus, and then those nuclei divide and get propagated through that haploid individual to make it a dikaryon. But this is one of the really cool options that these fungi have, because if they keep their nuclei separate, they can do this. If they're diploid, they can't easily do this. This is called a imon mating, where a dikaryon mates with a monokaryon and donates a nucleus to make the monokaryon. And we don't know how common this is, but I would guess it's fairly common, because when mushrooms are making spores, they're going through the air, landing on the ground, and some of them are going to germinate and come into contact with already established dikaryons of the same species. So that dikaryon can mate with the new haploid individual from the spore and make it a dikaryon, and then that can make its own little colony. So I'm guessing this is somewhat common, at least in some species, but it's something to think about, that the spores that are being made might be mating with established individuals rather than having to find another new partner from another spore. But if you look at the literature, I haven't dived into this, but there's a lot of research on variations on all these themes about tricaryons and the armillarias that are diploid and how they can exchange between diploids and haploids. A lot of different, more messy things going on with what these fungi are doing. The Heterobasidion, which is a polypore group, has been looked at a lot because it has some weird conditions in the hyphae where there's you can have cells with a bunch of nuclei of the 2 types, and they're not sure what's going on there. They're not just 2 nuclei per cell. This parasexual cycle is something I don't really understand completely, but it's a way to get, it's like a fake sexual cycle. There isn't any meiosis going on, but we do have some karyogamy type things going on. But we have 2 haploid individuals. They can fuse together and make cells that have nuclei from both individuals there, the heterokaryons in the top center. And then you can get karyogamy, fusion of nuclei, and we get some haploid nuclei mixed with diploid nuclei. And then, but the fungus wants to go back to haploid state because that's its basic living stage. But we don't have meiosis. What we have instead is, on the bottom left is number 5 there. We get back to the haploid stage by just throwing away chromosomes. So we have the diploid nucleus is going to have 2 sets of chromosomes, but to get back to haploid, they just throw away extra chromosomes until they're haploid. So it's strange and mysterious, but there's some people that think this might have been the earliest sort of pseudo-sexual life cycle for the earliest fungi. But there's obviously more research is needed for that. There's rust fungi. They're complex. They switch between different kinds of plants. In this example, this is the wheat rust. It switches between wheat and barberry and has 5 different types of spores, lots of different structures for moving between the 2 different plants and then going through the seasons. So if you want to get really complex, you can look at rust life cycle. Bird's nest fungi have splash cups where they shoot up little packets and they land on a plant and then dry out and open up and release the spores. So it's a way to get a package of spores up into the air and away from the cup, and then get it a little bit farther up into the air when the spores are released. So ascomycetes, we're going to go through that and see how those are different. These, in some ways, these are even more strange than mushrooms, if that's possible. And I don't quite understand why they do what they do, but it's what they do. So in this case, we're talking about morels and cup fungi and lichens and dead man's fingers and other carbon fungi and things like that in the ascomycete group. So the difference here is the ascus is the reproductive cell equivalent to the basidium, but the spores are made on the inside. And those ascus cells or asci are either in an open cup or a closed up chamber, depending on what group of fungi, what group of ascomycetes. So here's our basic dicarian life cycle, and what we're going to do is change some of the parts on the top. So the first thing that's different is these 2 haploid individuals, instead of mating and making a dicarion that lives a long time, these fungi live, these ascomycete fungi live a long time as haploid. So they're in the soil or the log or whatever as haploids, and they only come together when they want to make a reproductive structure, when they want to make a cup fungus or a morel or whatever. So they come together and they work together to make that fruiting structure, the cup fungus, which is what I'm showing the top center there, that N plus N in that little strange hemicircle thing. That's representing the fruit body that's made up of both individuals. And then parts of that, in a part of that, the 2 individuals fuse to make a dikaryon stage, and that stage then goes on to make hyphae inside that fruit body that then end up making the asci, which goes through the karyogamy and the meiosis to make the spores. So that's a bit different from mushrooms. So here's a cup fungus example. So this could be Aluria, the orange peel, or it could be the scarlet cup, something like that, or a helvella maybe. And you have the 2 individuals coming together. In this case, they're blue and pink. labeled there. And they both work together to make that the cup shape. And then they fuse. There's some of the hyphae fuse together to make the dikaryon hyphae. Those dikaryon hyphae then grow up through the structure to the surface of the cup where they make the asci, which are going to make the spores. So this is a morel life cycle, and I wanted to point out briefly that I figured out today, comparing this with this old life cycle with a newer one, this is actually wrong. So this, if you look carefully, you can see what's labeled N or monokaryon versus what's labeled dikaryon or N plus N. And it turns out that the morel life cycle does not have a long-lived stage. According to this new research, the morels don't have a long dikaryon stage like mushrooms do. The dikaryon stage is only in the actual morel mushroom fruit body. So this paper from 2017 looked at a bunch of black morels, and they found they looked for markers that tell you which tissue or cells have which mating type. So on the far left, we have a morel. That's a cross section of a morel mushroom, along with an ascus with the 8 spores, and that shows you where the mating types show up. So in this case, we've got a mating type labeled yellow and a mating type labeled pink. And on the example on the left, both of those individuals work together to have hyphae that make that whole morel structure. And the dikaryon hyphae go into making cells in the hymenium, which in the case of the morels is that orange part, those depressions in the top of the morel. Yeah, that's orange because that's the dikaryon, and that's making the spores, which go through meiosis. So you end up with the separate mating genes in separate spores there in the ascus. In the center, we've got a morel case that's not as common, where only one of the individuals is making the structure. The other individual, in this case, the pink mating type, is mating and fusing to make the hymenium, making the dikaryon stage, but it's not actually making the morel structure. And then on the right, we've got the odd case that's not common, where we have an unmated individual that goes ahead and makes a morel mushroom, maybe in the hopes that it'll, in the process, find a partner, but it doesn't find a partner, so it ends up making the morel mushroom just out of its own haploid hyphae. It's unmated, so it can't reproduce. There's no mating, and there's no possible spore production. So this is how you get a morel mushroom that is sterile. So if you ever find a morel that's not making spores, then you found the rare case where it did the mushroom. So this is their life cycle, and the dikaryon is limited to being inside a morel mushroom. And the overwintering stage and the sclerotium and all that other stuff, the mycelium, is all haploid instead of a dikaryon, which is what they thought before, because that's what regular mushrooms do. But anyway, this is quite interesting. Here's a lichen life cycle. I'm not going to explain all this. I was trying to show a life cycle that showed all the different moving parts of a lichen, because a lichen is part fungus and part alga. So you have the fungus life cycle on the lower right, you have the alga life cycle on the lower left, and then you've got the lichen reproducing itself on the top left, and you've got some mating things going on in the top right. So this is showing a different option. So if you ever want to lecture on lichens, I can recommend Matt Nelson. Can explain how lichens are different and how they're important and the cool things that they can do. Real briefly, in our last minutes, we're going to look at the other simpler fungi, those chytrids, zygos, and glomeromycota, and then a couple other things. So chytrids are either aquatic things, or some of them are parasitic, and they're either mostly single-celled or sometimes a group of cells that attach to something they're eating. So in the top center, those little pale circles are chytrid fungi that are digesting those pollen grains. And on the top right, we've got 2 chytrids on that algal cell. The damage, the really bad chytrid that's out there is the one that's forming the disease on frogs and other amphibians that's causing problems and stressing out the frog, causing population problems. So this is that chytrid fungus on the frogs, Batrachochytrium. Its life cycle is diploid and also asexual. Here's a different kind of chytrid that's grown for biology labs. It's got an alternation of generations. It's got a haploid and diploid stage, and both sides make different kinds of structures and spores and whatever to switch between the two. It's quite pretty for a chytrid, actually. Here's a zygomycete. This is the black bread mold. It has a haploid dominant. The only diploid part is the zygospore or the zygosporangium. There's a picture of that at the bottom right. Otherwise, it lives as a haploid mycelium. Glomeromycota are a really important group. They form the mycorrhizae on most plants. And we know the least about their reproduction because these spores they have are huge and have thousands of nuclei of all different kinds of genotypes. So we can't parse out whether sex is going on or not because it's not a simple case of 1 or 2 nuclei in there, so we can know what's going on. It's like a community of nuclei in each spore. So we looked at fungi. There's actually 2 fungus-like things that used to be grouped with fungi, but they're not related. There's the slime molds, and that is a group that's related to amoebas, and they're in the amoebozoa. And that group is related to the animal and fungus group there in the lower left. And then water molds are in another group on the top there, and they're related to a certain group of algae. But they're fungus-like because they have nutrition and they make spores kind of like a fungus. So here's slime molds. They have an interesting life cycle. The part that we usually see is the after mating. It's that plasmodium there in the lower right, and then we often see the spore-producing parts of the life cycle, which are making haploid spores. But this is a different kind of critter. The plasmodium crawls around and ingests bacteria for food. The oomycota are important plant pathogens, downy mildews and blights. The potato blight is in this group. And then there's a few aquatic sapro decomposer type oomycetes, like saprolegnia on the right, that you can grow in culture and show people in lab. And this is showing the egg. It's all mycotas named egg fungi, and this on the right you can see it's got these big egg-like cells in that structure. So now we're going to go over and answer questions. So do you want me to look through the chat first?
Sorry, I had to move locations, so I'm now in a place with really bad internet. So hopefully you can hear me.
Yeah.
Okay. Also in the dark. But I can read from the chat if you like. And anyone who wants to unmute yourself, you can either say so in the chat or just go for it in a lull. You can ask your question yourself, or you can put it in the chat. So I'm going to ask just one of mine because I actually did lose internet connection. So I'm going to have to ask you, Tess, or Rose, or Melissa, or somebody who's had the chat for longer because I blanks out when I lost internet. But I can just ask one of mine to get going. It's conceivably, could more than one fungal species be threaded together, producing fruiting bodies, not just with different mating types, but with wholly different species, spreading using the same body, fruiting body, as like the vehicle for that. Similar to a lichen, but just 2 or more fungi species with no of the algae. Or has that been seen, or
I
do
don't
you
know.
feel that it's
I
possible?
don't know any examples of that. There are some cases where one organism will hitch a ride on some kind of spore-like structure. The phycomyces are the Pilobolus thing. I think that's a zygomycete type, and that grows on dung and stuff and makes these little thread cells that stick up and make a swelling. And on the top, you've got the sporangium of spores. And it shoots that whole sticky sporangium off at once. So all the spores are shot off in one package. And little nematode worms climb up the stalk and hang out on the top. So when that spore package gets shot away, the nematodes also get shot away and get dispersed. But I don't know about 2 fungi that kind of intertwine and make the same structure and get dispersed at the same time.
Okay, I was just seeing in that photo or in the illustration you had of that morel, there just seemed to be, it just felt like there was an opportunity for that, the way you were showing the different mating types having different kind of segregated places
Yeah.
to express themselves.
So yeah, I don't know of any cases where 2 different species are joining together and doing anything.
Would really mess up taxonomy. Just like, but
Yeah, well,
okay, thank you so much.
Related question is, I don't know of hybrid fungi, or I don't remember examples of hybrid fungi, which are more common in plants and animals. And I think that's partly because fungus mating is based on the chemistry that's related to the mating types. So there's a chemical control on that. Whereas when you mate a horse with a donkey to get a mule, that's a physical process. Or like making other animal hybrids or making orchid hybrids, you're moving pollen from this flower to this other flower of a different species. And that's bypassing the natural process of having a specific pollinator do that for the flower. So you're getting around the way that organism is naturally doing sexual selection for a mating partner. So in some cases where things are related enough, where there's not genetic barriers, you can get a hybrid. But so I don't know. I don't remember any cases of hybrid fungi. I can check again with some other people because it's just based on chemistry.
Yeah, that's interesting. Also, that I could see why plants and animals would not have bothered to evolve such strict chemical controls if they have another mechanism for gatekeeping. Basically.
Yeah, I mean, they're basically these like specialized orchids and things where you have a very special flower for a special pollinator. The need for a chemical difference, I guess, has less pressure on it, so they kind of let that slide. But a lot of plants do have a chemical part, a process in when the pollen germinates, it won't be able to grow down to the egg if it's not allowed to, because the female part of the flower has to support that pollen tube growing down. So there's a chemical relationship there. And some plants can recognize themselves, so the pollen from the same flower from the same individual can't get down to fertilize the egg. So there's different levels of chemistry going on, but there's also cases where you can have related things mate.
Right, and then also what we might call more mechanical barriers.
Right. The biggest mechanical barrier is geographic isolation or separation, and you get a lot of speciation from that, just because the 2 organisms don't come in contact, and then they can both differentiate over time. There's only a couple questions in the chat here so far. There's some people that want a microscope, and I think you said that the club is going to be getting one.
We are, so And we are pretty much about to be doing that. And if you do, if anybody in this meeting, or obviously anybody who's a member, wants to reach out and discuss more about
Did I recommend?
We did. And
Okay.
we are pretty much, or I'm in the process of basically getting the microscope. And so far I'm going.
Yeah, I have it right here because we bought it for school and the school sent it to me because I can use it at home for my Zoom for my classes, and it wasn't going to do anybody any good at the classroom that's closed. So this is what it looks like. Whoops, I can unplug it. So this is a Swift microscope. It's a real basic, good quality scope with the objectives and condenser lens under there, and it's got 2 eyepieces, and then it's got if you get the one I recommended with the photo tube, and then you get the Swift Cam 5 megapixel to put on the top, and you're all set. And this plugs into your computer, and you can. I've been showing students stuff under the microscope during Zoom with this, and they really enjoy that, especially pond water stuff with critters. Last semester we saw tardigrades and amoeba and all sorts of things.
Yeah, so that's a great use for it. So I will emphasize we are getting a camera attachment for everybody who wants to suggest things, suggest topics, and at some point we will be able to meet more in person. And I could foresee us doing, do you want to get to know fungal microscopy? And it could before you make the investment, you may be getting a microscope of your own, or if you just want to be using it at club events, it's certainly a way to get into more of this like mating type and the budding and the clamps and, understanding, and really no better way than by doing. But we might present with it as well. So that was Hunter who was asking, or who thought, like, first of all, she said that your presentation was so cool. I saw that like a couple times in the chat, and also the microscope excitement. So that's great. And did anybody want to unmute themselves and ask a question or put it in the chat? I had another one in there, but I'll give privilege to anybody else.
Patrick, I was curious. I have heard that that Fundus is changing a little bit about what they're going to be doing DNA sequencing on. Do you know anything more about that? Or
No, I haven't really been following that. You mean how they sequence, or what they're going to sequence?
What they sequence.
Oh, okay. I don't know that. Are they going to focus on certain groups or something? Or what?
I think so. They're looking for more rare species, and they're moving away from just sequencing whatever people are sending in.
Well, that's tricky because you're not going to. I mean, in a way, if everybody did that and they thought all the chanterelles were the same, they never send in chanterelles. So you never find where all these chanterelles grow. So I kind of disagree with that, but I mean, you have to know enough to know that this is something interesting rather than something that you think is the same as something else. And that's in every group, like Pluteus and boletes, and I mean,
Well, and that's what I thought of in the very beginning of the lecture when we were looking at those chanterelles. That's what came to mind. And so then to follow that up, last month we had Will Padilla Brown, and he's set up his lab in Pennsylvania, and he's doing his own sequencing with one of these sequencers that he can take out into the field, I believe, right, Rose? I don't know if Rose is on.
Sorry, I was having trouble. I was having trouble unmuting myself. Yeah, I don't know. Well, he explained it in his lecture very briefly. I don't quite recall if he was doing all the sequencing or if he was just doing extracting and then like sending it to a lab. It wasn't 100% clear. I'd have to go back and rewatch it.
He's probably sending it somewhere because the last step of sequencing is you need a specialized machine for that.
Yeah, but he said he had gotten it down to 10 cents a sequence, which that just blew my mind when he said that.
Oh, really?
Yes, because he bought this $1,200 like sequencer that he can take out that's like portable.
Okay, well, I'd be interested to find out about that because
I know, so would we.
I don't know. I haven't heard of anything like that.
Because I was thinking if that's something that's really coming down the pike, maybe that's something we want to look into for our area.
so-
Maybe, we could, for
Right.
both of, for all of our stuff,
Yeah,
for Northern
you
Illinois and.
You
and-
need a dedicated person that knows how to do all that.
Right,
Yeah.
right.
Yeah, I could actually comment on that. He was using the Oxford Nanopore. He was using the that's the mini ion sequencer.
Let me write that down.
You want to put that into the chat, Randy?
Oh, sure. Yeah, sure.
Do you have a link for that?
I could get one quick. I'll put it in chat.
That would be great.
There was this little, I forgot what it was called, this little project on Kickstarter several years ago with it did the amplification, that first step, and some had a little centrifuge thing and stuff. So it allow you to get the DNA extracted. I don't know how well those are going.
So he's putting it into the chat now,
Okay.
Patrick. So, because we have at the university where I work, I've had some discussions with them about possibly creating a small lab area for us that we could maybe have access to. So that's like what I'm in discussion with them right now
Where was that?
at Concordia.
Okay.
It's in Mequon. But it would be, obviously, it could change. I mean, depending on where we'd want to put our things. But that's always an issue, is to have some place that we can go and do things.
Right. With the microscope,
the
it was
link.
like I was going to be holding it, but then it's like, where do we get together to use it as a group? And, if people have ongoing projects, that is so exciting to us. That's, like speculative. We're working on it, but
Well,
it's
yeah,
a
and
really
that's
cool
how
idea.
that's how these things come to fruition. We have to have the ideas first and sort of form them, and then start to look for the avenues of making them happen.
Yeah. If you don't mind, Patrick, I'm going to
That's
ask my
the-
second question
Yeah.
that I had.
Right.
I put it in the chat, I think, but I also copied it, luckily, because I can't see anything in the chat really from earlier.
Okay.
But it was, oh yeah, is there any rhyme or reason for spore shape? As in, so like studying pollen from plants that we were talking about, there's like those certain shapes of pollen that they can kind of, with evolutionary reasoning, pinpoint why it's that shape, that it's that shape for wind, water, pollinator purposes. Is the variation in fungal spore shape or structures on the spores similarly understood as being for that reason and patterns, or is it like unknown right now?
It's not well understood. I think some people have guesses. Recently, with lots of data, people are, I don't remember this correctly, but I think there's a very broad change in spore size by latitude, if I remember right. I'm not sure if that's what I remember right, but there are really, it's only mostly guessing at this point. The ones we are definitely sure of are aquatic fungi, have very weird spores with very elongated projections. And that allows the spore to tumble through the water, but get trapped in the leaf litter, so it just doesn't get washed downstream. So it gets tangled in the leaves, and then it grows on the leaves. So these spores are like anchors to get attached and not get washed away. But the spores that go through the air, they're different sizes and shapes and colors. And the deal with the color is a spore that has a heavier pigmentation, like brown or black, that's like sunscreen. So they can survive a little bit longer in the light when they go through the air. So white spored things, those spores, we're thinking more and more, those spores do not survive very long, and they're not going to go from America to Europe and survive that trip. They're going to go from, this part of the woods to the other part of the woods in the shade of the trees. That's what they're going to do. They're not going to get, they might not survive going from this patch of woods across the parking lot to the other patch of woods. Maybe, I mean, they'd probably survive that far, but they're not going to like go long distance in the sunlight. Pale spores, darker spores and thicker walled spores. Things like truffles, underground fungi, they make spores that are quite resistant and longer lived, and those spores hang out in the soil and can survive quite a while. It's like a seed bank for the fungi. But the spores that go through the air, people are discussing why the spores are different shapes. People have been doing experiments with wind tunnels to figure out different mushroom shapes. And the shape of the mushroom cap does have some impact on the wind currents around the mushroom. So the idea is that if you can disrupt the air current enough, you can make the spores get wafted up higher into the air. There was that news article which had a weird title. I think it was last year's, mushrooms make their own wind. Anyone saw that one? It's kind of a silly way to write it, but mushrooms can, because of the shape and also the release of moisture, can affect the air currents and help the spores rise up. There was a program by another mycologist we had with our club that talked about spore dispersal, and they've been doing work on these spores that are shot out of these cup fungi into the air. You've probably all seen that, where you can disturb a cup and it poofs spores up. Turns out that there's a couple things going on that are cool. So there's a whole cup with asci that are going to shoot spores, and that starts, one of those shoots spores, and then it causes the next one over to shoot. So there's sort of a wave of these asci shooting spores, and these spores are shot up, and that starts to make an updraft of air. So this first spores that are shot up start making the updraft, and then the later spores get caught in the updraft and go higher and higher. So the spores that are shot out later end up going higher into the air than the first spores that go up. So it's kind of a neat aerodynamic thing, which I didn't know about before.
Yeah, it sounds like a relay race, kind of.
Yeah,
Or like a team pushing each other forward.
Yeah, right. Yeah, so you can put slime molds on a naturalist, rust fungi, and other things.
Yep, and we have it's in the chat somewhere, but I linked to the slime mold project that WMS has, and so that's anyone who wants to join it can. Anyone who is in any of our projects, I add you to that. So any slime molds you see, we will all see, and it's a good way to get a sense of just what slime molds are, what they're about. And if you, as I noted, if you are going out looking for fungi, you're going to see some slime molds, which is a benefit.
Yeah, right.
But they're mostly amoeba, I think, if not all.
Well, they're slime. Well, slime molds are all, the true slime molds are in the amoebozoa group. There's different groups of slime molds, but we mostly see what's called the myxomycota, the big slime molds. There's really tiny cellular slime molds, but those are in the soil. They crawl around and make spores in the soil. It's not something we're going to see. But there's one little critters that grow in the lab, and they're called slugs. They're a smaller colony of cells that move along like a slug on the auger.
Oh, this was reminding me, because you said in the soil and something that we're not supposed to see. Would you say that to what degree?
Oops.
You muted yourself on accident.
She's not muted.
Yeah, sorry. I fell out. Sorry, Tess, you can go ahead.
No, I was just going to say you've had a little bit of trouble.
Yeah, with the internet. Oh, it was just basically, how much do you think lab artificiality goes into our difficulty understanding mating type sexuality in fungi? Or lab conditions versus what would be happening in the soil, or how directly are you able
Oh,
to
yeah, I
observe?
mean, if you study stuff long enough, you figure out that some of the things that the fungus is doing in the lab or whatever, it's probably doing that rarely in nature. But there's also cases where we can look at a fungus and in lab and whatever, we only see asexual reproduction, but we can look at the genes and see that it still has the genetic machinery to go through the sexual life cycle. So it either lost sex recently or it's rare and we haven't seen it. So there's a lot of mysteries out there, actually, what some of these fungi are doing.
Thank you. That is really interesting. And the point about using the genome to read what's actually possible for it,
Yeah,
potentially.
that's really cool. As we do more genome sequencing, we can actually look at all the different genes and figure out what proteins it's making and compare different organisms and figure out, like one area they're doing that with is wood rot, because there's a lot of different enzymes involved with wood rot. The other speaker you should have, if you haven't yet, is Dan Lindner from Madison. And he can talk about wood rot and some very interesting things with that. He's a guest in my class on Zoom, and he's done a couple talks for our Illinois club. But and of course,
Maybe we can talk, and Patrick, let's talk in the next couple days, and I'll get some contact information from you. And I had some other questions too I wanted to ask you outside of the meeting. Absolutely.
Any questions on reproduction and fungi?
Any other questions at this point? If not, I'd like to remind everybody that Patrick's going to be up at Hiles. I did in the chat put in 2 articles that he mentioned about the air currents. And I also put in the Wisconsin Mycological Society's event page. And on the event page, there's links to all of our events that are coming up. And some of those events you really don't want to miss. One of them, of course, is going to be Hiles, which is July 22nd through July 24th. There is a foray fee. Those fees go towards the mycologist. The mycologists go up there early and they really look for where you're going to have the best experience, the most interesting experiences, as well as looking for edibles. So, the money that you spend on those forays, really, nothing really goes to WMS. It really is forwarded over to them for their travel and, a daily stipend, as well as a place to stay. So, we're really supporting our scientists this way. We also have the NAMA foray coming up in Estes Park in Colorado, which will be August 12th through 15th, and registration is here. And our board member, Rose Tercy, will be there. There's a lot going on. It's really going to be fun. The Telluride Mushroom Festival, I think, Britt, Pat, are you going to be up at Telluride?
No, I'll be at NAMA.
You'll be at NAMA. So definitely, there's 2 places where you can go and talk with Patrick.
Or the Northwoods foray in September is a very good foray up north.
And that is going to be an amazing foray. In fact, WMS is really taken on as a community partner to work with Fungi Magazine, Britt, and with Patrick to make that foray happen this year. So again, there's watch your emails. There is reserving your spot, and also the Lakewoods Resort is on the events page that I put in here. And again, one of the things that we're going to do, we've talked with Britt about it. So if you, reserve your spot through WMS, there will be WMS only forays up there that Britt and others will lead. So there's going to be a women's only foray. There'll be a couple of other forays only for WMS members. So really, this is an important thing for you to do. And the only way you'll find out about them is exactly the same way that you find out about our lectures. We'll send you an email with the foray time and meeting spots. Also coming up is the Mycelium Mysteries, which will be September 24th, 25th, and 26th. And we also are supporting them this year. So, we're sponsoring part of their event. And then Mitchell Park Domes is going to do the show dome about mushrooms, and we are the community partner for that. And so in that case, I've been working with some of our board members that we've been making mushroom sculptures. They're going to be all around the domes with mushrooms, mosses, and mums. And we will be there, and so will Mushroom Mike. He'll be selling mushrooms, so foraged as well as cultivated. And that will be on September 26th at Mitchell Park Domes. And then the Fungi Fair is coming back. Yay! And it's going to be at the Polish Cultural Center, and it's going to be amazing. Mushroom Madness, October 9th. And so a lot going on between now and the end of the year. So definitely check out that events page. Go to some of the events. You know, WMS really appreciates you and your membership. And I did put, we did create an ad for NAMA this year to help support NAMA. And we did talk about the Northwoods Free in there. So check out the ad. It's on the chat. Yes, Rose.
Tess, you should screen share it because it's just showing up as like a download in the chat. You should screen share that beautiful ad.
All right, I'll screen share it. Hold on. I've got to go find it now.
Don't also forget that we have the rustic camping foray coming up at the end of August, like right before school starts, and people who want to, you know, just start learning, it's going to be a really, really good beginners event. So that's coming up as well. It's our very first camping foray.
So there it is. There's the ad that we'll be putting into the Northwoods to NAMA, to the North American Mycological Association.
It's a full 2-page ad that's going to be in the NAMA program.
Wow.
So
And
we're
I
also
really-
representing Wisconsin.
And I really love the albino deer. Because, you know, we've got our albino deer up by Buffalo County. So the annual Northwoods Fray, and then of course, information about the Mycological Society. And all you have to do is click on the QR code. It'll take you right to the events. So pretty exciting. And we're super, super, super excited to be able to help support the Northwoods Fray and make sure that it does not go away, because, you know, it's such an intense, wonderful fray. It's probably the best North American fray. So I really encourage everybody to.
It's the best one in the Midwest for sure.
I think it's the best foray in the country. It's got more, I think there's more fungal diversity there than anywhere else.
Well, we don't know yet because we haven't identified it all yet.
Yeah.
Ha ha ha.
That just means we got to study it more. And don't forget, guys, we have a YouTube channel, and we're going to put some lectures on the channel. Some of them are going to be private because we're going to re-air them at later dates. One of them is up there right now. The Morel lecture is public. And the iNaturalist roundtables we've been doing, I'm going to start posting those after I edit them. And lots of fun little short videos, little mushroom spotlights, little foray spotlights, and they're all very educational.
Yeah, and if you have an idea for a video or something you're really passionate about, definitely contact us with the email or on the website.
Yeah, you can do it on the website just through contact, or just send an email to the Wisconsin Mycological Society, the ones that you get all of your info from.
Or you could send a PM to our Facebook page too, or Facebook or Instagram.
Yeah. And I want to say thank you so much, Patrick, for coming and presenting. Also for all the work you do on your website.
Thanks.
It takes a lot.
I haven't done much since this winter, so.
But see, that's still a lot. That's what I want to say. It's still, even if it's unfinished, still a lot.
Yeah. I've only got 200 pages and there's 1,000 fungi, so.
Right, only 200.
On the other thing I'm doing is each time I want to add a species page, like the chanterelle, I have to make a genus page, a family page, an order page, a class page. So what I did a couple of years ago was make all the pages for the orders of mushrooms. And then I made all the families for polypores, which is one group I want to fill in more. I'm not doing as much with guild mushrooms. I'm doing some of the odd non-guild mushrooms, I want to sort of complement Michael Kuo's website in a way. I don't want to duplicate everything he's got. Eventually, I want to like have a framework with links to his stuff, but I don't necessarily want to duplicate the species he has. I want to put some more unusual or some less less studied or less charismatic stuff like crusts and sterium and polypores and stuff. So
Right, and that would
one of the
very-
other new pages I added was for the glue crusts. I did research on that and realized that one of our common crust fungi is actually a glue crust, and it's like my goal to actually find it gluing branches together. There's only a couple people with pictures of it doing that. Gary Linkoff has a picture of it. But up north, there's another glue crust called alder glue that glues alder branches together. We
Wow.
found that up there a couple of years ago.
real quick, so is this only at the upper branches?
No, it's branches that fall down. Oh, no, it's branches that are still on, dead branches still attached on the tree or the shrub. So what this fungus does is it grows on a dead branch, and if it comes in contact with another branch that's touching the first branch, it'll grow and colonize the other branch, and the mycelium, by growing it over both, glues those branches together, and that prevents the other branch from falling to the ground and getting eaten up by something else. So it's a way to kind of hoard dead branches up off the ground.
Oh, that's that's pretty ingenious.
Yeah.
So it keeps its food source away from other
Yeah,
possible
and in the tropics, there's
material.
marasmus-like little fungi that grow up and capture the leaves before they land on the ground.
Very cool.
Yeah. But
Well,
thanks,
we-
and I'll see you up in the woods in Wisconsin.
yeah, can't wait. Up in the woods soon. So
I'll
good
see you
night,
next week at
everybody.
Hiles.
Yep.
Have a wonderful evening. And again, thank you so much, Patrick. We always enjoy having you here.
Okay.
Yeah, we have a
Well,
lot of thanks in the chat.
Yeah. Thank you.
All right. Good night, everyone.
Take care.
Thank you. Good night.
Thanks, Patrick. Good night.
All right. Good night.